everyone. Our next topic is blast and substitution matrix. Okay, so let's begin with the class. So, um, what is BLAST? So, BLAST is a sequence similarity search program for comparing biological sequences such as the amino acid sequence, or you can say like nucleotide sequences of different protein of different nucleotide of DNA sequences with sequence database or library sequences. Okay, so what is doing BLAST is, suppose you are having a query sequence. Okay, so what query sequence can be either a protein or can be a DNA. Okay, so it will search against any kind of sequence databases, like if you are having protein sequence, it will search against the protein database or the nucleotide database, or if you are having the nucleotide sequence, suppose you are having here protein sequence, then it can search either nucleotide database or can by protein sequence. If you are having nucleotide sequence as a query sequence, then it will search again the protein sequence or by the nucleotide sequence. Okay, in either way it can perform the search. Okay, so what is BLAST? BLAST is B stands for basic, L stand for local, A stand for alignment, and S stand for search, and T for tool. So basic, local, alignment, search tool. Okay, so it will identify, or it will predict the similar matches against the databases. Whatever databases you will provide as a query, it will search against those databases. Okay, so in the BLAST there are different kind of BLAST, basically amino acid sequence BLAST or DNA sequence BLAST. Okay, it is categorized into two different type of BLAST. First one is the amino acid sequence BLAST and DNA sequence BLAST. Okay, so in amino acid sequence BLAST there is two type of BLAST. First one is the BLAST P and second was the T BLAST N. Okay, in DNA sequence there is BLAST N, BLAST X and T BLAST X. Okay, so here we will see, like in this column many questions have been asked in previous year questions, so we will see this column very carefully. Okay, so if you will see the first one, that is the BLAST N. Okay, so the program name is BLAST N. Okay, it is a kind of nucleotide or DNA sequence BLAST. Okay, here you are giving a query sequence as a nucleotide sequence. Okay, so there are two boxes there in the BLAST program. Okay, two boxes there. So one is your query sequence you have to provide. Okay, as a input in FASTA sequence you have to either provide the nucleotide sequence or you have to provide the protein sequence. Okay, so this will be your target sequence. Okay, so against which this BLAST can be performed. Okay, so the first one is the BLAST N. So in BLAST N what will be happening here, the query sequence is nucleotide. Okay, this nucleotide either can be DNA or can be RNA. Okay, it will search against what kind of database? So it will search against the nucleotide database. Okay, in the previous slide I have told you earlier that the nucleotide database can be either searched against the protein database or nucleotide database itself. Like that, protein query sequence also searched against the nucleotide database or protein sequence. Okay, so here the program is BLAST N. So BLAST N query sequence is nucleotide and database is the also nucleotide. Okay, the second one is the BLAST P. Okay, this is the simplest form of the BLAST. Okay, so BLAST P, here also query sequence is protein and database is also protein. Okay, so here in case of BLAST N both the thing are nucleotide. In case of BLAST P both the thing are protein. Okay, things are protein. So here if you can if you want to remember, like N denoted nucleotide, so both the thing is nucleotide. P denoted to protein, so here both the thing is protein. Okay, so then you are having mixed blast. Okay, so mixed blast, the first one is the blast X. Okay, so blast X, how you will remember, like blast X is you are having the nucleotide sequence, but you wanted to search against the protein sequence. Okay, so in this case the query sequence will be your nucleotide. So this program, what it will do is it will convert the nucleotide into protein sequence by considering all the 6 ORF. Okay, reading frames. Okay, open reading frames, it will consider all the 6 open reading frames, and then it will translate into the protein, and this protein sequence again search against the protein database. Okay, so here your query sequence is nucleotide, but you are searching against the protein database. Okay, that is that thing is done by blast X. Okay, the next one is the T blast N. So T blast N is just opposite of this blast X. Okay, so T blast N, what it is doing is, T blast N basically here you are having the protein. Okay, query sequence is your protein. Okay, so and here you are having database is translated nucleotide. Okay, so here you are searching query sequence as a protein, and against whom you are searching the translated nucleotide. Okay, so it will again translated nucleotide means it will again means it will be like protein sequence only. Okay, but the databases which is having the translated nucleotide. Okay, it will perform the blast against the translated nucleotide. T blast X is both the things are translated. Okay, and both the things are nucleotide. That you have to remember. T blast X is both the thing is translated and both the thing is nucleotide. Query sequence is also nucleotide and the databases is also nucleotide. Okay, that you have to remember. So how you will remember these things? So first one is the blast N, simply N denote for nucleotide, so both the thing is nucleotide. Okay, so blast P is basically P stand for protein, so both the thing here is protein. Okay, blast X is X is here translated nucleotide. Okay, so when you are having blast X, that time what is happening, the translated nucleotide is searched against the protein database. Okay, so that nucleotide is firstly translated into protein, then protein query sequence is searched against the protein databases. Okay, T blast N means here you are having the protein sequence and you are searching against the translated nucleotide. Okay, that will be also protein. Okay, so but it is present in the form of translated nucleotide. Okay, then you are having T blast X. T blast X is translated nucleotide search against the translated nucleotide. Okay, translated nucleotide, both the thing are the translated and both the thing is nucleotide. Okay, so this is the different type of blast. So in 2018 there was one question asked, which one of the following blast search program is used to identify homologue of a genomic DNA query in a protein sequence databases. Okay, here it is asking which blast search program is used to identify homologue of a genomic DNA as a query sequence. Okay, genomic DNA as a query sequence in a protein sequence database. Okay, genomic DNA as a query sequence, and you have to search again the protein sequence. Okay, so here we have seen that blast P, both the thing are protein, so it will be not the correct answer. Blast N is also protein, so it will be also not correct answer. Okay, here blast X will be the correct answer. Okay, because here we have seen that blast X is translated nucleotide which is searched again the protein query sequence, protein databases. Okay, and the next one is the T blast N. T blast N is the your query sequence is protein but your databases is translated nucleotide. In the question it is not like that. Here your query sequence is genomic DNA. Okay, and you have to search again the protein sequence database. So the correct answer of this question will be blast X, where the genomic DNA is as a query sequence and protein sequence as a databases.